Hello, everybody. It's my pleasure to welcome André Antunes from um, the University of Science and Technology in Macau, in Asia, to this next seminar of the European Astrobiology Institute. And actually, André is coming from a university city, namely from Coimbra in Portugal, which is one of the most important universities in uh, Portugal. And he has actually also done his undergraduate studies in biology there. And after doing his PhD at the University of Regensburg in Germany, has been quite a lot at quite a lot of places. For example, at the University of Coimbra, at the University of Cabo Verde, at the King Abdullah University in Saudi Arabia, at the Edge Hill University in Ormskirk in the USA, until he joined the Macau University of Science and Technology, where he is associate professor now and leader of the astrobiology unit and lab coordinator. And today, the theme will be about astrobiology in Asia. And welcome, André, and the floor is yours.
Well, thank you very much. Thank you for the introduction, and thank you once again for the invitation. It's a pleasure to be talking to a different audience and tell you a little bit about my research. So let me just move to the next slide. A little bit of information about us, starting with my career path. We already got an overview here. I worked a little bit all over the place, which is more or less normal when you pursue a research and career in research and come from a relatively small country where there aren't that many opportunities. So I really did go to a lot of different places and had wonderful experiences everywhere that I went to. I've been here in Macau for 4 years now. I came a little bit before the pandemic, so the timing was a little bit off, but I've really been enjoying the time here. Regarding the group, the astrobiology group that we have at the State Key Lab, this is the only Chinese State Key laboratory for lunar and planetary sciences. And we recently established this Astrobiology Research Group, and the idea is to promote this research field and sort of kick it off in China because it's still new. It's a new thing here in China also. So these are the current faculty members that we have working with different aspects of astrobiology. And here is an overview of our postgraduate students. Some equipment highlights that we have. And this is sort of a general research topics overview ranging from limits of life to astromycology, applied space microbiology, looking at biosignatures, for example, and development of new tools. I'm going to mention some of these things during the presentation. Okay, so starting with life on the edge, and a big part of the focus of this presentation. So I'll be focusing more on some of my research background and research experience and the research that is done in my team. And of course, we are all aware of this now, that we live in a microbial world. Microbes have been able to populate every single corner of our planet, no matter how extreme. And there's a range of different extreme environments that were previously thought as inhospitable to life, that likely were sterile. They were even declared sterile at certain periods in history. They actually host magnificently diverse microbial communities that are very, very successful. And we have the examples that I have here are extreme pH conditions, for example, in the top left corner, extreme salinity in the bottom left corner. And then we have examples with high temperature, for example, both surface and deep sea locations. So a big part of my research career and a big interest in my team revolves around a very specific type of extreme environment. It's actually one of the most extreme environments on our planet. And these environments are called deep sea brines. The deep sea brines were discovered completely by accident in the 1950s. It was a Swedish research vessel that was collecting samples. They were doing a transect across the Red Sea and collecting samples at different points. And one of the samples that they recovered came back to the ship, and people were surprised that it was actually quite hot and quite salty. And so this marked the discovery of the very first deep sea brine. There was a lot of interest on what was happening here in the Red Sea area. Nobody really understood how could this be that you'd have at the bottom of the Red Sea these locations that had higher temperature and extreme salinity, and this was something quite unusual. So there was a big, massive survey that looked into different parts of the Red Sea, trying to understand this phenomenon a little bit better. And so we currently know have identified around 25 different brine pools in the Red Sea region alone. Later on, they were also identified in the Mediterranean region, and there are also some equivalent type of environments in the Gulf of Mexico, although they're a little bit different. What is it that creates this? Like, why is it that they are there? Why do we have deep sea brines? Basically, they are formed by the tectonic rift that occurs between, well, for the Red Sea, the tectonic rifting process that occurs between the Arabian and the African tectonic plate as they are splitting, and the sea is opening up, and the new ocean is being born in this area. And this occurs by deposition of new oceanic crust. This occurs at very specific points along the central axis of the Red Sea. And the deposition of new oceanic crust, because oceanic crust is denser, that means that this process of deposition of new oceanic crust generates a series of deeps, of topographical depressions. So this creates the deeps. Where does the brine come from? The brine comes from also from the tectonic movement and the rifts that are created as a part of this, the fractures that are created in the subsoil. And what we have underneath the whole of the Red Sea area is the remains of a prehistoric sea that completely dried up. So we have this massive layer of concentrated salt, of halite, that is found in the subsurface. It's found underneath the bottom of the Red Sea. And because you have this tectonic movement and the creation of these fractures in the soil, this allows normal Red Sea water from the deep to trickle down, to go through the cracks, have contact with this partly exposed salt layer, redissolve it. And at the same time, you have hydrothermal activity, so you have increase in temperature, input of heavy metals. So there's change in the chemistry, and there's a very high increase in salinity. And so as the seawater resurfaces, its chemistry has changed and its salinity has changed. That's how you have the formation of brine. Because brine is much denser than seawater, its tendency is to sink. And what happens is the brine is created, it becomes automatically trapped in the brine pools, in these topographical depressions that were formed. And that's how you have, in a nutshell, that's how you have formation of the deep sea brines. Despite the fact that they're all formed in a similar way, you actually see big variability. So here I picked two of the most extreme examples. We have Kebrit Deep and Atlantis II. Atlantis II is the biggest one. It was the first deep sea brine that was discovered. Temperature keeps on increasing, and temperature is currently around 70 degrees Celsius. So it's the hottest one. It's the biggest one, a total surface area of around 30 square kilometers. And Kebrit Deep is located further up north, so it's more recent. Hydrothermal activity hasn't really kicked in that much, so temperature is much lower. It's around 24 degrees Celsius, a little bit below 24. And it's one of the smallest ones with a total surface area of around 1.5 square kilometers. In addition to this, we have input from local geological layers. So each deep sea brine has a unique combination of chemistry and physical conditions. So you have differences in temperature and difference in concentration of different compounds, making these environments truly unique. An additional aspect that is really interesting about deep sea brines, because brines are so dense, normal particles that would fall throughout the water column all the way from the surface and would normally go all the way to the bottom. When they reach the brine body, because the density is so big, particles tend to accumulate at the brine seawater interface. It acts as basically as a particle trap. So you have this accumulation of very high density of particles that occurs at the brine seawater interface. And this transition zone is very, very thin. So you have within the space of a couple of meters, sometimes as narrow as 2 meters, you have drastic transition, drastic change of conditions, where you have increase in temperature, increase in salinity, depletion of oxygen. And this is illustrated by the picture that I have here, and one of the latest expeditions that I participated in, where we had an in situ filtration system, and we see 2 filters there, one that looks orangey and one that looks dark brown. This is filtered samples that were taken within the brine seawater interface, and the distance between both of them is around 1 meter. And you see that chemistry is really very, very different. So even within each brine, depending on where you're sampling, you have unique type of setting. To illustrate further this accumulation of particles that occurs at the brine seawater interface, we have this video footage from a ROV that was collected from one of the brines from Kevrit Deep. And you see this white hazy material that is accumulation of particles. We also identified specifically in Kevrit this thin orangey layer that was easily disrupted when we approached with the ROV. We think that this might be some sort of biofilm structure, but it's still not really clear what it is. It's something that we're keen on exploring in further cruises. So looking at microbiology, the brines were discovered in the 1950s, as I mentioned before. In the 1960s, there was this big campaign to get samples and do chemical analysis. There was big interest in trying to determine if this might be connected with oil presence, possibly. And so associated with these, we had the very first microbiology studies. And this is what they found. Absolutely nothing. The scenario was so bleak that the papers, I actually have the papers from back then, actually declared the brines to be sterile. So it was thought that conditions were too extreme for life to subsist. It was a combination of multiple extremes that no organism would be able to survive. And truth be said, now we understand this a little bit better. The techniques that were used back then relied on standard cultivation using Petri dishes, and it's normal that you're not able to capture the full picture regarding microbial diversity. This is a well-known documented phenomenon. It's usually referred to as the Great Plate Count Anomaly. And even if you just look under the microscope, you see a lot more morphological diversity than the number of different colonies that you observe in Petri dishes. This is a very well-known fact. It comes from a variety of different factors that I won't really go into now. Microbiology has changed since then, and we now have this sort of more holistic perspective and holistic approach using a combination of collection of environmental data, cultivation-based approaches, and molecular-based methods and combine data from different types of sources to get a better picture of the full scale of microbial diversity at any specific environment. So the first phylogenetic studies that looked into these brines came much later, much later than when compared with the first microbial studies. And what they've shown was that actually there were a bunch of different brand new phylogenetic groups present in the brines. So rather than being sterile, you actually had very complex microbial communities that were specifically adapted to conditions, to the conditions that you have in the brines. They were completely new and unrelated to, they didn't really have any closely related sequences from other environments. We had further studies later on, including studies that were done in deep sea brines of the Mediterranean that kept on further expanding the new sequence groups that were unique and were directly associated with deep sea brines. And we had some really interesting analysis that confirmed that microbial community structure was layered, where there was at the brine seawater interface, you had transition occurring within the microbial community, which makes sense because you have complete shift in environmental conditions. So this was reflected in microbial communities. Regarding microbes that were actually isolated and described from these places, I'm not really sure what type of backgrounds I have here today regarding audience. I just wanted to show this picture so that you have a better notion of the different taxonomic levels in an easier way to understand, all the way from domain, which is the higher ranked taxa, all the way to species. And so this was the very first microbe that was isolated and described from this deep sea brines, called Flexistipicynus arabici, and it represented a new phylum. It was actually isolated from Atlantis too. And so a new phylum would be equivalent to finding the very first chordate. So that was a very important find. This microbe and a few of the next ones that I'm going to mention are actually isolated using alternative technique called optical tweezers, which relies on a laser microscope and allows us to bypass the need for Petri dishes and makes it easier for us to capture more of microbial diversity that and escape the Great Plate Count anomaly. The following one is Sanisphera japanensis. So this was the first one that I worked with during my PhD. It represented a new order. So that would be equivalent to finding the very first carnivore. Sanisphera japanensis was isolated from a brine seawater interface, had remarkable metabolic versatility, physiological versatility also. And so this flexibility that we found seems to be or should be an advantage in such a harsh environment. So it was able to grow with oxygen, without oxygen, microaerophilic conditions, with pressure, low pressure, different ranges of salinity. So it seemed to be really flexible and adapt really well to the drastic change in conditions that you see at the brine seawater interface. We also sequenced the genome later on and found a variety of genes that explained or helped to support why this microbe did so well and adapted so well, including a couple of genes that could be interesting for biotechnological applications. But the most interesting of all was this specific microbe here called haloplasma. And so just to give you a little bit more context, particularly if you're not a microbiologist, if you look at the microscope and see microbial cells, they usually come in one of two basic shapes. They're either spheres or cocci, or they are rods that look a little bit like baguettes. And that's the two basic shapes that you normally get with microbial cells. There's some additional variability, but 95% or so of all cells have one of these two basic shapes. So if you see something that has a different morphology, you automatically know that there's something interesting going on. And this is the case with haloplasma. So haloplasma has this central round body and has these projections, these tentacle-like arms that coil and uncoil. And this is something that is unique for the bacterial world. And so unique morphology, unusual cell contractility cycle that we observed. We also did phylogenetic analysis, so we were able to confirm that it was also quite novel. It represented a new branch between the firmicutes of the mollicutes. These are two big groups of microbes, and seem to be some sort of missing evolutionary link between both big groups because it shared some properties with one of the groups and some with the other. We also did some scanning electron microscope analysis to try and get a little bit more information of the complexity of the shape of the cell. And this is a really interesting microbe that there's still a lot of questions that we haven't been able to address. So there's still a lot of things that can be done with it. Another microbe that I wanted to highlight is Halorhabdus tiamatea, represented a new species. The reason why I'm highlighting this one is it was the first, and for a long time, it was the only member of the archaea isolated from deep sea brines. It seemed to have a widespread distribution. It was detected in other sequences related to this were detected in other deep sea brines. And also from a metabolic perspective, it's the only member of the haloarchaea to have a preference for growth without oxygen. So this was something that was quite unusual. Also isolated some curious microbes that precipitated biominerals, which is something I'm not going to discuss now, but I will mention a little bit later on during the presentation. Okay, so I'll return to this topic. So an additional highlight, I wanted to mention that it's not all about microbes in the deep sea brines. We actually also find some more complex life forms, and we were very lucky as part of one of the expeditions that we had, one with ROV. We were able to get samples from the shore of the brine and collected these mussels that turned out to be a new genus of mussels. So we're also quite happy with that find. So at this stage, you're probably thinking, oh, this is really interesting, and the Red Sea sounds pretty cool and has some really interesting microbes, but how is this all linked with space? So you're probably aware of this. Astrobiology is a new research field, or relatively recent research field. It's cross-disciplinary. It brings together research from several different fields of expertise and different types of techniques. A big focus of astrobiology is the search for life outside our planet. And so normally people refer, when they try to gather some sort of description for astrobiology, astrobiology is the study of the origin, evolution, and distribution of life across the universe, or simply speaking, trying to look for life outside our planet. That's an oversimplification. Of course, it's a little bit more complex than that. And regarding the search for life outside Earth, big focus is on Mars. And this has been since a very long time. Mars has a long history of capturing people's interest. Part of it comes from Percival and Lowell's drawings of the surface of Mars and made it quite popular. And the theories that this might be some sort of artificial structures that were built by some sort of doomed civilization that existed on the Red Planet. Of course, this fueled a huge wave of creativity from popular culture, ranging from books to TV series, movies, different forms of science fiction, even music was inspired by it and continues to do so up to today. Of course, with the first missions that went to Mars, people lost a little bit of interest. Everybody was sort of expecting us to find little green men, and that didn't materialize, and Mars seemed a lot more inhospitable than was originally thought. The public opinion shifted elsewhere, and people stopped caring so much. We are now returning to Mars quite in force. Part of it has to do with this mantra from astrobiology, follow the water, and the fact that we have more and more evidence supporting that liquid water might be possible on Mars, on the subsurface, and possibly even on the surface under certain conditions. So all life as we know it requires the presence of liquid water. So people are, again, really, really interested in studying Mars and trying to understand whether life might have existed in the past or might actually still be there. And of course, Mars has been capturing all of the attention, but a big focus will be moving on also towards the icy moons of the outer solar system, places like Europa and Enceladus. And the reason why these icy moons have been getting more and more attention is the fact that there's evidence for global scale oceans. And these global scale oceans likely interact with a rocky core, possibly with salt. So there's a lot of interesting things going on. There's still a lot of unknowns because we have thick layers of ice at the surface. So it's more complicated for us to understand what's going underneath it, but there are different models that try to estimate what type of chemistry could we expect in these oceans in Europa and Enceladus. What we do know is that, or what we expect is some sort of localized hydrothermal activity and higher temperatures, high pressure. Chemistry from Europa is not so well known. From Enceladus, we know a little bit more. part of it, what we expect here is also high salinities in certain parts, high pressure. pH is likely alkaline in this case. And the reason why we know more about Enceladus than Europa is the fact that Enceladus has these plumes that project from one of its poles. And you actually have material from the ocean being sent into outer space, which should make it much easier for us to get better insights in what's happening underneath the thick crust of ice. So when I mention Mars and Europa and Enceladus, people usually just assume that regular people think that I'm going to work with samples from Mars, or that I already have them, or that I have some samples from these oceans from the icy moons. Unfortunately, that's not the case, not at this stage. So a big focus of astrobiology needs automatically to be on our own planet. Luckily for us, our planet has a variety of different extreme environments, and some of them actually are not that different from extreme conditions that you find on other planets. So the reasoning is, if you find life on extreme conditions on Earth, why not find them also on extreme conditions in other planets? Here I need to introduce the concept of terrestrial analog. So terrestrial analog sites are locations in our own planet that have a variety of conditions that are similar with conditions in other parts of the solar system. They're very, very useful for astrobiology. So they allow us to test the limits of life, namely looking at polyextremophiles, their source of microbial strains for testing and exposure. They allow us to look into long-term viability and preservation, both of microbes and biomolecules. And they also allow us to test technology, develop new technology for space missions, both for sampling and for simulation. So before we actually send new missions to Mars, it's normal to head to one of these terrestrial analogs to do some testing. Regarding the specific case of the icy moons and the oceans of the icy moons, we have a tricky situation. What we're looking for is multiple extremes, and it's not always easy to find locations that match this type of multiple extremes that we're looking for. So basically what we're looking for are brines with varying chemistry, high pressure, and some sort of hydrothermal input. Luckily for us, that's exactly what the Red Sea offers, and deep sea brines in general offer. So that's why we recently proposed it as an ideal candidate for terrestrial analog site for exo-oceans. And so there's a lot of interesting things that can be done from an astrobiological perspective. Also, planetary protection is an important aspect. Planetary protection is a vital thing. It's basically what planetary protection consists of is a set of rules to ensure the preservation of future conditions for future biological exploration. And on one side, protecting our planet from contamination coming from other planets. So if we collect samples from Mars, making sure that we have proper containment measures on one side, but on the other side, also make sure that when we send missions to locations like Mars or the oceans of the outer solar system, that we're not contaminating it ourselves. This would be and that's a serious risk that we incur in. There are regular discussions on planetary protection measures and any adjustments that need to be done or should be done. I was actually involved in some of the discussions on the planetary protection of the outer solar system linked with the icy moons, and we got some really interesting publication out of this. And this was a joint discussion that involved all major space agencies. There's also very important discussions on, specifically on the exo-oceans, on the need to do more experimental and simulation efforts. And we had this group effort that was published in 2020. We're very happy with this because it was picked as a Nature 2020 Research Highlight of the Year. It's one of the top 6 papers of the year for astronomy. And specifically with planetary protection, it's very important for us to have access to model organisms, to reference organisms, and see how exposure to space conditions affects them. What is their survivability, what is their capability to grow and to thrive and survive space exposure. Salnisfera chabanensis, which was one of the microbes that was isolated from the Red Sea, the first one that I worked during my PhD. We had a partnership with DLR in Germany. We performed a variety of stress tests looking at desiccation under anoxic conditions, radiation, Mars-like atmosphere, and we have a couple of ongoing experiments, but we recently published it as a new reference organism for planetary protection perspectives. This microbe was also sent to the upper part of the Earth's atmosphere. It's planned to be sent to the Antarctic for exposure experiments there. Currently in discussions sending it on a satellite. So in addition to that, it's been selected as a group, as part of a group of microbes that will be sent to the International Space Station next year. Our results so far have been really, really interesting regarding tolerance to desiccation and tolerance to UV. And for the next part of the talk, I already mentioned a little bit the let's follow the water mantra. I want to focus on something a little bit different and offer a slightly alternative take. Let's follow the salt. So water is essential for life as we know it, as I mentioned before. All organisms on our planet, all life as we know it, depends on the existence of liquid water. That's the reason for the mantra to begin with. And so when we look at Earth, there's plenty of liquid water. So the link is clear. But when we look at Mars and look at what Mars looks nowadays, and with it being so dry and barren, the link might not necessarily be there or might be as immediate. But if we look at Mars in the past, we actually know that there's evidence for existence of high quantity of liquid water. So looking back at our own planet, what happens when seawater evaporates? It produces salt. Salt precipitates. And salt has been, on one side, used historically for preserving food, preserving humans even with the mummification process. And so it has been used traditionally as a way of protecting against decay and sort of preventing the growth of microbes. But the existence of high levels of salt is actually does not oppose the existence of microbes at all. What we see, for example, in coastal environments where salt is traditionally produced is these flat solar salterns where water turns pink or red. And what actually this indicates is very high salinity and very high density of microbes. You see a shift in color associated with shift in microbial communities. The big champions here are members of the archaea, specifically the group called halobacteria. But they are representatives of all three domains of life that thrive under high salinity conditions. Artemia is an example of animal, Dunaliella, microalgae. We have bacteria, Salinibacter ruber, Halobacterium and Haloquadratum are examples of archaea. And they all do really, really well. There's a variety of different high salinity environments, including salted foods, the salters that I mentioned before, deep sea brines, salt mines, for example. So there's quite a wide range of different settings that we could look into. When seawater evaporates, the salt is produced, and this is mostly halite, sodium chloride, both in quantity and volume. So what happens to the microbes that live in salterns when the salt precipitates, when water is completely gone? Is this the end of the story? So could it be that microbes are trapped in there and sort of dying off, trapped and buried alive inside crystals of salt? It's actually not what happens. They are trapped inside salt crystals. We can actually sometimes even see them having this sort of reddish, pinkish coloration, when you have a high density of microbes trapped in there, for example, halophilic archaea. But what happens is if you put them back and mix it with water and redissolve it, you release the microbes again, you release the archaea again, and they're able to grow, they're fine with it. A study has looked at 14 different types and tested it for 6 months, and they were all fine. Looking more in detail, we now know that microbes survive because they are trapped inside brine inclusions, these small bubbles of water that are formed inside the salt. These can be very, very abundant, so you can have very high number of inclusions. Apparently, they're able to fulfill some sort of basic metabolic needs by recycling organic material from cells that die in there. So it's sort of like a small microcosm type of thing going on. Some microbes reduce their size, have a variety of adaptations that allow them to enter this sort of minimal metabolism stage and keeps them going for a longer period of time, keeps their viability. We do see some changes in diversity that we still don't understand very well, and it depends on what types of microbes are trapped together. There's these cross-linking happening between different microbes that are really interesting. Interesting aspect here from an astrobiological perspective is that survival within these crystals might enable some halophilic archaea to survive over geological time. So this would make halophilic archaea the oldest living organisms on Earth. There's some controversy here. There have been some issues with the very first report. People were very skeptical about an isolation of a 250 million year old halotolerant bacterium from a primary salt crystal. So this was a very big find, but was heavily debated whether this might be some sort of external contamination, whether they were really trapped or not. But people kept on reporting across the globe similar findings using the best techniques possible to make sure that there was no external contamination. Current record seems to be at around 830 million year old fluid inclusion. So this keeps on stretching. So that's really interesting. There's a bunch of different microbes that we know that have been detected in these brine inclusions and are capable of long-term survival. There are additional benefits here from a Martian perspective. Microbes that are trapped in salt are somehow sheltered from more extreme conditions, namely from exposure to radiation. They're basically in a golden cage. They have organic material, they have water, they're more sheltered, so it's actually not a bad thing for them at all. So regarding Mars, what happens? Like, what's the setting over there regarding salt deposits? We do know that there are chloride deposits on Mars. So they're halide-bearing deposits in at least 640 different locations. They're relatively easy to access, and I think they're really useful resources because they would trap organic material, water, biosignatures. So these should be targets for future sampling missions. Of course, there are still a lot of unknowns. All of the studies that we have tend to be focused on salt production on Earth and on Earth conditions. So there's still a lot of questions that need to be addressed regarding the specificities of Mars and the production of salt crystals in the Martian conditions, and how that might eventually affect the viability of microbes for geological times and Martian settings. So when discussing Mars, I think that following the salt might actually be a good option. Moving on to big data, but still staying on this topic of high salinity and organisms that live under high salinity conditions, we are very interested in extremely halophilic archaea. And it's something that we wanted to explore further. We do see that there's some big issues with this current age that we're living in with big data. It's incredibly difficult to navigate it. We have more and more data being generated, and it's more and more difficult to actually get full picture. So I already mentioned before that regarding life under high salinity conditions, which is directly relevant for discussion of life on Mars, extremely halophilic archaea are the champions and should be a key target group. They're recognized as a very important group for astrobiology. And so this is how the HAPPY project was born, trying to collect information from all types of metadata for all species description for all members of this group. And the idea was to build a database, an online tool that people could use to filter out information, quickly look for and find target species that were able to grow under specific settings that we were interested in. As a parallel project to this, we were also interested in looking specifically at what type of astrobiological testing had been done before. And this is work from Jiahui Wu. And what we found is, despite the fact that these are recognized as very important for astrobiology, regarding exposure-based testing, there are huge gaps in coverage. There are some really good examples with some species that are where we have very good coverage, but not complete data sets, and other groups within the halophilic archaea where we have basically no testing done. This evolved further, and so this tool is about to be released online. This evolved also to a parallel database looking specifically at the bacillus group and production of biominerals that I'm going to mention in the next section, and also future target biomining, specifically biomining linked with space exploration. Our new tools are very valuable resources. We already identified target for future studies, and the work with bacillus group will also be really important for in situ resource utilization, which is what I want to mention now. So microbes are incredibly important from an application perspective. They're increasingly seen as the big solution to all of society's current big issues, big problems: how to feed the world, how to fuel the world, and how to heal the world. Extremophiles and microbes from high salinity environments are used for a very wide range of applications, from biofuel, new drugs, bioremediation, bioplastics. There's a very wide range of things that you can do with it. Also, specifically, or a more exotic type of application is the production of biominerals. There are several different groups of microbes that have the capability to precipitate minerals, and we were specifically interested in carbonate biominerals. So microbes producing minerals is a really interesting thing to do research on. On one side, you're bridging the gap between bioscience and geoscience, and this is also always a really interesting thing to do, connecting fields that are frequently apart. From an application perspective, production of carbonate biominerals has been extensively used now for civil engineering, for repairing concrete cracks, for example, repairing works of art, improving quality of agricultural soils, and there's even some interesting pilot studies on CO2 capture. Concrete is an immense thing. The scale of production of concrete on our planet is just mind-boggling. And it's amazing how energetically costly it is and how it's responsible for a big chunk of CO2 emissions. And it's something that we normally don't hear about. So there's definitely demand to try and improve this. Again, what's the link with space? The link with space comes from this question that I've had since I was a small kid. Why don't we have a base on the moon yet? The issue here is that each space launch costs millions of dollars. Currently, all the material that we use needs to be sent from Earth, and that's a complicated thing. Costs and volume of heavy cargo are a major limiting factor. So there's this increased drive towards something called in situ resource utilization, which is transform, extract value, extract resources, transform what is readily available on the surface of other planets. And that could be lunar dust, for example, lunar regolith, Martian regolith one day. The idea is to try and reduce the amount of things that we need to take with us. So our motivation, we are interested in situ resource utilization. Microbes are seen as a major player in this specific aspect. So you have microbial in situ resource utilization. And our focus then is on microbial calcium carbonate biomineralization. Why? Because on one side, it's already a more or less mature technology on our planet. It's a good proxy because it relies on direct contact between microbes and extraterrestrial material like lunar dust, for example. And strains that are good producers of carbonate biominerals are also frequently used for other types of things in biotechnology. So they are very, very good proxies. So unfortunately, there's still a lot of unknowns regarding production of carbonate biominerals, and the feasibility of using microbes to produce biominerals and consolidate lunar dust to produce some sort of lunar bricks has been speculated, has been proposed, has been seen as very high potential, but people haven't really analyzed if this is going to work or not. So we definitely need more work. Bacillus is the key genus here. But there's, as I said, there's still a lot of unknowns. First part, what we wanted to do is compile information about all the genes that are involved in carbonate biomineral production or had been reported. We looked at the bacillus group and generated these genomic fingerprints that gave an overview of the types of genes that were linked with calcium carbonate biomineral production. And what we saw was that there were formation of specific subclusters. There was some variability that more or less matched well with phylogenetic positioning. Looking at this and in the lab also, we also saw that production of biominerals also seemed to be not exactly the same across all subclusters, but so closely related species produces minerals that were more similar than species that were further apart. So we do see some interesting links here that we're exploring further. We're also able to use these genomic fingerprints as a way of predicting whether a species that has never been tested actually produces it or not. And this worked really, really well. We also got some good results regarding yields, the amount of biomineral that is produced, and some good candidates for future use. We are now doing some tests on actually using this and testing whether this is going to work with lunar dust and lunar regolith. So we acquired lunar simulant material and used it for a set of experiments using 2 different bacillus species and using a variety of different techniques. The good news here is cells kept on growing. So both species that we tested grew well, regardless of the concentrations that we were testing, and kept on producing biominerals. Curiously enough, when we combine exposure to artificial lunar soil and microgravity, what we see is that simulated microgravity seemed to have a positive effect on production of biominerals, but concentration of the simulant had opposite effects in both tested strains. So this is a really interesting aspect. And it raises questions on in situ resource utilization. The best suited species for application on our planet might actually not be so good when you try to apply them to a lunar condition. So we definitely need to do more intensive testing and be very, very careful with what type of strains or species we're using or planning to use for in situ resource utilization. So an additional aspect here, I wanted to show this picture. So this is from Junjie Zhang, a postdoc that recently started in my group. We have ongoing experiments in actually trying to produce these lunar bricks, and we've had the first case of success of actually consolidating using carbonate biomineral production to consolidate artificial lunar material. We have a series of upcoming tests, so watch this space for new results. Some other directions for the final part of my presentation, because I know that I'm running out of time. I just wanted to mention a couple of new sampling campaigns. The big focus has been on deep sea brines, but we have a couple of projects that look at salterns, high salinity sites in other parts of the world. Big expedition associated with the Antarctic with partners in Chile. I'm scheduled to travel to the Antarctic early next year and get some new samples from there. And we have some new deep sea samples. Martha from the astrobiology team has just returned from a 1-month-long expedition to the Red Sea, where she collected new samples from the deep sea brines and also from some hydrothermal influenced areas of the Red Sea. So we're really excited to see what we're going to get out of that. New research direction that we're exploring now is how production of salts and precipitation of salts under Martian conditions might affect long-term viability of microbes. I mentioned Martha before. She works specifically with astromycology, so looking specifically at fungi and more particularly filamentous fungi or molds. This might seem at first glance a little bit unusual, but this is a group of really interesting organisms, really interesting microorganisms that can be seen as friends or foes. And it's very important for us to understand how exposure to space-like conditions might affect how they interact with us and how we can use them for different types of things. As part of this, she was successful on a recent project application that received support from United Nations and the European Space Agency. As part of this, part of our team went to the Netherlands and had access to hyperjets, and so we were able to perform experiments where we exposed filamentous fungi to hypergravity condition, 10G and 15G, using the large diameter centrifuge at the European Space Agency facilities in the Netherlands. So this is something that took place during the summer, so we're also really excited to see and analyze the results and the data that was generated out of this. Finally, to mention work from David. David works with biosignatures and preservation of biosignatures in extreme conditions, namely acidic conditions like in Rio Tinto, Spain, and preservation under hyper-arid conditions with collection of samples in several locations across the globe. So wrapping things up, I wouldn't have been able to do this work or show the results that I've shown you here without the support of several different institutions and funding agencies that have been very helpful throughout the years. And as a final shameless way of getting attention to this, I ask you to come and join us if you're interested in coming and joining us as a student or staff member. We have a variety of different research groups and research directions. We're expanding. We're still relatively new. Macau is a really amazing place, mix of Eastern and Western influences, Portuguese and Chinese. So it's a really nice place to live in and work in. The university and the State Key Lab have been doing really, really well, and we've been going up in the international rankings. Thank you very much. I'm now available for questions.
Okay, thanks a lot, André, for this overview about the deep about life in salty environments, and especially in these brine layers. There is already the first question by Jordan Stone, and the question is, what progress, if any, have you made to build international collaboration on astrobiology space missions between China and leading organizations in astrobiology, just such as NASA and ESA?
Okay, that's a great question. Well, we have made some progress. I must admit that we've had some limitations due to the pandemic. Keep in mind that I moved to Macau 4 years ago, so it was just a little bit before the pandemic, and this is a new research field. We had to set up the labs from scratch, so we wish that we'd be at a more advanced stage by now. Regardless of that, we've been doing really well regarding international collaborations. We had a very good link with DLR, for example. I mentioned the project with hypergravity that was United Nations and European Space Agency. Regarding the link with NASA, that's a little bit more complicated. Not necessarily from our side, but my understanding, and this is the information that I get from friends and colleagues that I have in the US, there's an embargo of sorts for collaboration between people working with funding from NASA and researchers in China. So the barriers from the American side, not from the Chinese one.
Yes, there will be other questions. Sebastian Fellner, please.
Sorry for the complication. First of all, great work, really nice. And I hope that I will be able to formulate my question right, because I love this astrobiotechnology. First of all, the wording of itself, but also wondering why don't we have a base on the moon, right? So like there should be one. I'm completely on your side. But I was wondering, especially when it comes to microgravity, because I've conducted like some experiments with foraminifera, like active biomineralization, like sent to space for 90 seconds, right? So like, but to have an idea if there's a blueprint of biomineralization, which is basically gravity dependent. And so I was quite interested by your results that showed that gravity obviously doesn't make such a big deal when it comes to biomineralization of the microbes, because also when you look at cancer cells, for example, they behave quite differently when they send to space, right? So they cluster somehow. And I mean, in long term, when you use them, for example, for biocement, or when you use them to really attach towards surfaces, or like also like to adsorbent surfaces and so on, I mean, I do think, or like I would assume at least, that gravity is a crucial factor when it comes to that, right? So like, what's your opinion on that? And could you elaborate a bit on those findings?
Well, so that's something that we're testing now. So the first stage was move from the theoretical side to actually the practical one. And so there have been a couple of papers that suggested, oh, this has high potential and this could be used. But people haven't really, there hasn't really been the experimental focus on using microbes to produce biominerals and see if it actually works under moon-like conditions. So that was the first thing that we were trying to test first, confirm whether known biomineral producers, what type of effect does microgravity have on them? And it's a weird thing. You'd expect that at this stage we'd have better understanding of what are the effects of microgravity in different living systems, but it's still a big unknown. Some species seem to be stimulated, others it's the opposite effect regarding their bioactivity. There's still a lot of unknowns. So what we did at this stage was pick 2 key species just as a sort of first test and see, are they affected by artificial lunar material or not? Are they still able to grow? Does this affect their biomineral producing capability? What if you combine both at the same time? And what we saw was the effects aren't universal. Even when we're talking about the same genus, 2 species within the same genus, for microgravity, you have one effect. For the artificial lunar material, you have the opposite effect. So we need to be very careful when picking specific species and see what's going to happen to them. At the same time, the next stage, which was what you were asking about, the production of lunar bricks, that's something that we're optimizing now and testing and seeing how will that affect, because of course it's different type of setting. If you're actually trying to produce and consolidate lunar material into proper bio bricks. But we're still at early stage. So these are just still preliminary results, but we're really excited because these things need to be tested and they haven't been tested so far.
Absolutely. And I mean, then next step will be Mars base, right? So then considering halophilic, maybe biomineralizing organisms that could also tolerate perchlorates, right? So I mean, that would be a completely different mineralogical setting when it comes to resources to be used.
So part of the thing I also mentioned, I didn't go into detail because, well, time is limited. And so I sort of opted for giving sort of a panorama, an overview of different things, and focusing a little bit more on some of the background and some of the things that we focus more on, but just provide sort of a brief overview so that people have an idea of different type of research directions that we do. But for example, regarding the database work, one of my students is working on this biomineral database with bacillus, and that's exactly the point. Once we have the database up and running, we can select what species should be the best suited for application on Mars or the Moon. So we can specifically look and filter out and just pick bacillus species that grow under specific salinity settings, or bacillus species that have been previously tested for growth and have been exposed to space-like conditions, so radiation, for example. So this is something that then, because there's hundreds and hundreds of species of bacillus, it's very time-consuming. We cannot test all of them in a short period of time. But it makes sense for us to try and prioritize and say, this is a good candidate, that's a good candidate, that's a good candidate. Let's test and see like is this going to work? Particularly when we, the little that we know and the little that has been tested regarding carbonate biomineral production is usually done with a handful of species, because they work really well and things are just optimized for species A, B, C, D, and E. And so people don't really test or use the others. And the most likely scenario that we already see just from testing these two is that it's very likely that the ones that work really well under Earth conditions are not going to be the best candidates for using them on the Moon or Mars.
Yeah. Nice. Thanks a lot. I'm really looking forward.
Thanks for the question.